And thank you for the Cancer Center leadership for giving me this opportunity to share my latest work. I have been working, I think my entire research career for almost 15 years has largely been on cancer, but the presentation I'm giving today, it's not much about cancer and not much about the single cell analysis I've been working on for almost 10 years. This is something we haven't published. It just came out in my lab. I'm happy to hear feedback from you guys. I think largely the unmet need in the omics area recently is so people can do sort of single cell omics and the multi-omics to really understand the tumor heterogeneity at unprecedented level, but you really don't have the spatial information anymore. So the spatial omics kind of came out or emerged to address this challenge. Over the past couple of years, I think largely you'll see many different technologies, but largely they are all based on just a FISH. The more specific, more precisely speaking, a single molecule level FISH. So the shortcomings here using FISH is it's difficult. Like even my lab working on technology, I just cannot do. This requires very advanced imaging technology, single molecule fluorescence. You need to image over sometime for very sort of high volume and genome scale data you want to collect from one sample, you probably need to image over days repeatedly to get this sort of large number of genes analyzed on the same sample. And also that's not a sort of unbiased genome scale. You really need to know the sequence you want to analyze. And also, so far, I think no one else talk about spatial omics, and the only terminology people use in this field is spatial transcriptomics, because that's pretty much they can do. It's not so obvious you can how you can extend to other omics measurements using the FISH. So I think the latest breakthrough came out actually this year. The two papers published, I think one just came out last week on Nature Methods. Another paper a couple of months ago, in Science, to really use the power of next generation sequencing for spatial omics mapping or spatial transcriptome mapping. So the approach they took actually quite similar. So they create sort of the barcode surface using the packed beads. So whoever working in this space probably know no matter 10x genomics on the homebrew, the DBiT-seq technology, you need a DNA barcode beads, so each bead has distinct DNA barcode to really tell you which messenger is from which cell, or they're from whether or not they are from the same cell. They're basically packing the beads in on a monolayer on a glass slide, and they need to decode the beads, need to know which bead has what sequence. So this decoding process was done by either solid sequencing or, again, very much like a FISH, you do repeated sort of hybridization and imaging to decode the beads. That is very tedious process as well. And but afterwards, you get a sort of freshly micro-sectioned tissue sample and place on top, and you lyse the tissue section, and hopefully the messenger is released from the cells sort of in the proximity of the specific bee should be captured only by the bee, but I don't think the lateral sort of diffusion can be really avoided. But at least they saw a pretty good preferential capture of the messenger from the adjacent cells. I think this technology published earlier this year in Science demonstrate you can do 10 micron resolution, the spatial mapping of the MRI transcriptome by sequencing. And this paper came out last week, demonstrate you can actually use even smaller beads, like a 2 micron beads, to further sort of reduce the pixel size and increase in the resolution. But 2 microns is really subcellular. The data analysis becomes even more complicated, and it turns out they have to be multiple beads to get a quality image. So interesting, when we visited their data, we found although they can see sort of anatomic or histological structure of different cells in the tissue, but it is almost impossible to visualize individual genes because the number of genes they can detect per pixel is extremely sparse, about like 100, 200 genes per spot. If you try to image all the individual genes across all the pixels entire tissue, the data totally sort of not that meaningful at all. So what we can do is fundamentally different. I'm not able to say too much in the technical details, but this is totally different. We don't use beads, and we just need a bunch of reagents and with this device. And although we have been working on microfluidics for years, but I don't like complicated microfluidics like you guys. So this device, you just place the PDMS on top of your tissue and you clamp it. That's everything you need to do to deal with the microfluidics. Afterwards, you just pipette your reagent to the host. So the data, the validation data we have shown is we use sort of the pan messenger RNA fish to visualize the individual tissue pixels. We eventually are able to sequence with the spatial resolution. So we found we can get a very nice 10 micron pixels as shown here if you zoom in. And also interestingly, we saw sort of the tissues after we process with our barcoding strategy, our barcoding approach shows some topological features. Even under optical microscope, you can see where your individual pixels are located on the tissue. And worth noting, so this is sort of exactly the same tissue we're going to take for sequencing rather than the previous methods. They always have to compare to the adjacent tissue. They are not able to get any good image from the same tissue at all. Also, the tissue sample we analyzed, they are just a peripheral, they are just a formaldehyde fixed tissue sample on the glass slide. So if you have a freezer of those samples banked in your tissue, in your freezer, we can look at those samples as well. We don't have to use sort of frozen tissue block and a fresh section and put on our slide. So we did some quantitative analysis of how many cells we can get per pixel using just a DBiT staining, and also we were also concerned whether or not each pixel is distinct molecular barcode. We can put on or some certain diffusion between the pixel that might cause cross-contamination, and we quantify the diffusion distance. We found using the fluorophores basically. So we found the diffusion distance is actually just a 1 micrometer, which means which suggests we can potentially further reduce the pixel size and increase the resolution to about like a 2 micron using our technology. So the feature size and match the sort of the microfluidic design very well, and the number of cells we can get in the 10 micron pixel size device is about 1.7 cells. We're really getting close to single cell level spatial omics. So as I kind of alluded a little bit earlier, so the quality of data very important. So we compare our data to the SLICE data published earlier this year. So the number of genes they can detect per pixel and about the same size, 10 micron, and then the number of genes we detected using our technology is really orders of magnitude increase in terms of how many genes, how many transcripts we can detect. About 3 years ago, similar technology, sort of the barcode surface based capture of messenger RNAs for spatial transcriptal mapping was published in Science 2016, but that was very low spatial resolution, about 150 micron. But in that data, when you look at how many genes they can detect, that's about the same as what we can do. But the resolution is much, much lower, or if you calculate sort of the area per pixel, it's 100 times larger than what we have. So I was very excited about this sort of data quality, which really enable on the following slides, we can really visualize individual genes rather than using extremely sophisticated informatics to identify gene set to just visualize the different cell types. We can actually interrogate every single genes across the entire tissue map. So when we first start with this, I'm extremely excited about the tumor microenvironment for sure, but we decided to pick something that's so well characterized, people know what cell types are there. So we use the mouse embryo in the earliest stage of the organogenesis. It's about 10 days. We were able to map out, actually I want to talk about a messenger, and actually we can do also about 22 sort of tens of proteins simultaneously mapped out using the same barcoding strategy, microfluidic barcoding strategy. As shown here is a sort of PAM messenger RNA, but done by sequencing. So you can see actually the intensity of the total signal of the messenger does reflect sort of the density of the tissue on the embryo slice. And here, the average signal of over 22 proteins we're able to look at as a panel. That doesn't really correlate that very well, but I think that makes sense because you're not looking at a global at all the proteins, but the subpanel, it really depends on what proteins you put in your panel. So then we did a clustering analysis. When we look at single cells, we use t-SNE, but here it doesn't make sense. You have to use t-SNE because you know exactly where the spatial location of every single pixel is. But the computational algorithm for clustering is identical. So, but after clustering, we just put it back to the tissue histological. The spatial map, we found we see sort of about 8 clusters over here, and the pretty much match the anatomic annotation we got from the eMouse atlas. And more interestingly, I think in the eMouse atlas, you are now able to kind of resolve the one stripe of tissue here, but we saw a very distinct stripe of sort of cell type. We were still unclear what those cells are, but probably associated with the mouse sort of major aorta around the area. As I mentioned, we are able to visualize individual genes or individual proteins at a very high quality across the entire sort of tissue section. Showing here a couple of genes and cognitive proteins. And overall, I think the protein signal way higher. It's not a big surprise just because you measure only like a 22 and rather than genome scale. But when you compare, you see consistency, you see concordance or also discordance between the gene and proteins people have seen over and over. Very interestingly, when we look at EpCAM, it's a very nice concordance between the protein and messenger RNA and the EpCAM expression right here. And this one, I think this microvasculature, so microvasculature already developed in a mouse embryo at this stage all over the whole body. We can see they are expressed everywhere, but we don't see distinct structure at this resolution because this resolution is about 50 micron, now 10 micron. I will get down to the high resolution data later. And then we did a sort of validation to compare our data to immunofluorescence staining for several selected genes. And there's a vasculature again, you see indeed extensive everywhere. You see EpCAM exactly the same pattern as we saw using sequencing. So just a couple of those locations showing the expression of the EpCAM. And another validation is we got the sequencing data and a paper published earlier this year by Jay Sendhu from University of Washington. They use single cell sequencing to map out several mouse embryos over different stages. And then you can basically do tissue, a sort of sample t-SNE or sample UMAP. This is not single cell UMAP, but a sample UMAP. So we found the 4 samples we sequenced actually mapped very well to this sort of differential or developmental trajectory. So in here, this is from their data, this is sort of day E9.5, and this is E10.5, and we're right in the middle. Those are kind of a little bit later stage of the developmental mouse embryos. And then we use a little bit higher resolution to look at the embryonic brain. This is about the entire brain and a little bit other tissues in the head and the neck. And also this one, we didn't know what that is, but after data analysis, we found actually it's a piece of the heart. What we see from the protein and from messenger RNA, so again, the messenger RNA atlas does reflect in the tissue histology very well, and the protein now at much higher resolution than 25 micron, you do see some sort of correlation between tissue histology and the protein expression atlas, but not as so distinct compared to the messenger RNA. So we were able to visualize individual proteins, and show here 4 of them I think are very interesting. Again, the epicam, this is a very high resolution. You can see very tight clusters of epicam expression in specific tissue regions right here and here, and this 2, 3, or 4. And a microvasculature, we can see the microvasculature by sequencing very well. And when you go to look at the tissue histology, or maybe I'm not pathology by training, I just cannot identify where the microvessels are located. Based on the tissue histology. And the 2 other proteins very interesting as well. There's a metacam we found is highly enriched in part of the forebrain, but not entire forebrain. And within the CD63, it's widely implicated in the early stage mouse development. It's kind of anti-correlated with the metacam in other areas. So we kind of put them together. You can see their relative correlation among each other. So again, this technology we really want to validate to make sure what we saw using sequencing does match the immunofluorescence staining. So this is from sequencing. This is from sequencing. This is about microvasculature. This is epicam. This is immunostaining. You see almost a perfect match. I was very surprised. This is really a perfect match. Distinct clusters right here. A little bit right here from immunostaining, and we can pick up. It's only a few, so 1 pixel, single pixel layer thickness, we can pick up very well. And so now here you can see those microvascular network using immunostaining, which was also observed in our sequencing map atlas. So I got interested in this particular protein called a metacam, and asked my postdoc to do some differential gene expression sort of, but the metacam transcripts is difficult to see the distinct, sort of spatially distinct expression, but the protein data you can see very well. Then we decided to use our sort of high quality spatial protein data to guide the differential gene expression across the entire transcriptome for different tissue regions. So in this case, we look at the metacam positive and the metacam negative, and map down the top ranked genes for metacam positive region. It's still ongoing since I'm still in the stage of learning developmental biology, but we can see some interesting features. But in the negative region, clearly, so this is hard, turns out. This is kind of hard, kind of microtubule associated proteins. And this is interesting thing. We don't really see this protein showed up extensively in the brain, but somehow look like in this local area. And I have no idea what that is, but later we figure that's actually the eye. Yeah. And then we decided to do even higher resolution, which is 10 micron resolution mapping of a particular region of the brain. And again, we had no idea where to map now. We just randomly place our device on top and then mapped out this region. And the red color actually real data. This is basically just a PEM messenger RNA data. You can see the signal relatively uniform and now perfect, but that's totally okay. Just like we do single cell RNA sequencing, we always do normalization. Then that gives you, as long as your sequencing data quality the number of genes you can read out, thousands of genes, you can always do normalization, compare across different pixels. As I told you, actually we can see in the same tissue, sort of after the barcoding and before the sequencing, we can even just under optical microscope, we can see individual pixels over here. And then when my postdoc showed me this image, I thought, okay, you got a king white fiber over there. very likely because we saw this when we used microfluidics before. I thought that's unfortunate, but anyhow, let's go ahead and process the sequencing data. But turns out that's not a kinwise fiber. That's really a very thin layer. It's actually a single cell layer of melanocytes lining around the eye field. At this stage, the eye field actually is a very, very early stage, only called eye vesicle, and even no eye optical cups. It's an optical vesicle. So we can see very distinct group of genes strongly enriched inside the eye and also lining around the eye, optical vesicle, and when we put them together, a little bit more structures you can see. For example, in the PAC6 enriched pretty much in the entire eye field, but also in this region, this optical nerve fiber. And but here, this protein only expressed in the eye, but also other tissue type, but not much in the optical fiber. You can see this very well at a very high resolution. It's really about a single cell resolution. So when you look at carefully, you see some yellow spots over here. That means the PAC6 and the PML actually are co-expressed in those kind of melanoblast cells, but this one is not. The 6/6 is not expressed only within the eye optical vesicle. If you further zoom in, you can see the sort of the gene expression within the vesicle and also individual pixels, every little square here. So we can overlay the tissue image and the transcriptome data. So we notice one gene which is strongly enriched right here, very strong differential expression spatially. We're curious what this gene does. We did a sort of this time it's still global gene differential analysis. We saw on the top ranked the genes, and these two showed up, but we found their function in on a top ranked pathways to some degree, okay, except those ones to some degree are mutually exclusive, and later we realized that, but that has never been observed before. I don't have sort of literature data to support, but it seems like those cells controlled by this characterized by this particular gene later on going to determine the development of the lens, and those cells even at this stage you don't see any morphological difference. They already predetermined to develop the retina and the photoreceptor cells. And then we were able to basically just pull out those pixels and compare to those to perform a differential gene expression analysis, and not a surprise at all. This gene just showed up extremely differentially expressed, and but we see many other genes. Interestingly, we're still very interesting. We're still trying to look into the details. So they are kind of enriched on the left side. Eventually, very likely. They will contribute to pigment, the photoreceptor cell development. Okay, so even though we were able to visualize individual genes, we don't have to use gene set enrichment to identify different tissue types, but we had a challenge, in particular in this kind of eye field region, due to our lack of knowledge in mouse embryonic development. But it would be great if some computational pipeline can automatically identify different features, tissue features. That's what we demonstrate as well. So using this automatic automated feature identification pipeline, we're able to identify actually 20 different features in this very small region of the brain associated around the eye field. I just show you some of those. You can see not just the eye, actually can see very early development of the ear based on the sort of gene expression, but histologically you cannot see any difference at all. We also look at the entire mouse embryo at the E10. We're able to identify about 20 different features, but we're asking so if at later stage, many other organs begin to develop, whether or not this pipeline can identify many more tissue features or tissue types, subtypes. That turns out that's right. And using E12, we were not able to cover entire embryo. Actually, just a lower part of the body, we identify about 40 different features already, and so this at a very high resolution as well. Okay, I'm gonna just summarize back to my the main interest in cancer. So I believe this enabling platform, we demonstrate we can do protein and the transcripts, but actually in my lab, another postdoc working on high spatial resolution epigenomics. I believe we can do high spatial resolution ATARC, high spatial resolution DBiT-seq, and the application extremely broad, and the cancer is put in right in the middle because that's really my main focus. I would like to thank people in my lab who work on this, and thank you for your attention.